Hello, I am Professor Sims, and this video is an introduction to the online microbiology lab. This course is broken up into 10 lab sessions, and each lab session has specific learning objectives. It is an online-only iteration of a traditionally on-site course curriculum. Teaching will be asynchronous, meaning that students will work at their own pace to complete procedures and assignments before established due dates. Lab sessions will not be live, but lab procedures should be performed on or before the dates listed on the course calendar, which is in the syllabus, in order to allow for enough time for obtaining observations and reporting your experimental results prior to your assignment due dates. Students will engage in self-directed learning, but you are encouraged to ask questions via Moodle discussion forums, email, and by utilizing the Zoom office hours. Learning will be assessed with online learning assignments, quizzes, and exams. Check the course calendar and the syllabus for the due dates, and check the syllabus also for a lot more details regarding what all is involved in these assignments. The learning objectives for this specific presentation and unit include becoming familiar with course expectations, resources, and information. Students will become familiar with the provided lab kits, including materials used for executing lab procedures, and we will discuss proper biosafety rules and disposal procedures. That is, you'll be briefed on how to properly dispose of used specimen plates and slides in a home lab setting that is safe and up to OSHA and environmental codes. The overall learning objectives for completing this course include becoming competent in bright field microscopy, microbe cultivation, learning the biochemical properties and microbial control methods, identifying unknown specimen. This lab is essentially a long-form supplemental instruction for the micro lecture course. In the lab, students have the opportunity to participate in authentic hands-on experiments, even from home. Such experiments include staining, culturing, isolating bacteria and other microbes, performing identification testing on microorganisms. Microbiology is a challenging field of science, and therefore this is going to be a challenging course. It focuses on comprehension, not just memorization. So you're going to be challenged to do your own literary research. You're going to be challenged to draw your own conclusions and interpret data, and you're going to have to learn how to effectively communicate scientific information, which can be challenging as well. Your learning is going to be self-directed, especially since it's an online course, but also because you're not only going to be learning about microbiology, but you're going to be learning a little bit about your own learning style as well. I encourage students to study smarter, not harder, by which I mean you want to get more out of your study time and you want to learn more in less time. So what I see a lot with my students over the years, I've seen them using these techniques here on the right for studying, things like rereading, rewriting, going over old quizzes, going over old class notes. And that's fine, except it's very, very time consuming, and at a certain point your brain kind of burns out, especially if you're doing so much rereading and rewriting and rewatching that you're losing out on sleep time, because your brain needs sleep in order to form new neural connections and memories. So I would like to encourage students to kind of shift away from these methods here on the right and shift towards these ones on the left, which have been scientifically proven to not only take less time to do, but they're more effective as far as long-term memories and comprehension of material is concerned. So you are going to do some rereading and rewriting, but what you really want to do is fill in the gaps of your knowledge. You need to kind of take inventory of what you do and don't understand, and spend your time focusing on the things that you need more help with. So you're going to write things in your own words. You want to test yourself, like write little mini quizzes and tests for yourself and answer them. And you want to try to interleave things. So you want to try to relate the new things that you're learning to things that you've already learned. And you also want to vary your studying in such a way that you're not spending huge blocks of time doing one activity or studying one concept. So you don't want to spend, 4 hours at a time just reading the textbook because, well, about 20 minutes into that, your brain kind of shuts off. You want to take breaks. You want to alternate between reading activities and writing activities and maybe other types of activities like watching a video or a simulation, or maybe if you're good at drawing and you're a visual learner, maybe you want to make a diagram or a figure or something like that. Anyway, you want to switch it up and make sure that you're studying smarter. Here are some resources and information that you'll need, and these are going to be provided in much more detail on the Moodle site and also in the syllabus. The textbook for this class is a free online OpenStax book published in 2016. Here you can see the link where you can go and download it in its entirety. From the table of contents in this book, you can click on any of the chapter titles. They're all hyperlinks. So if you you scroll to the table of contents, click on chapter 9. It will take you directly to the start of chapter 9. And that way you don't have to scroll 900 pages to get to chapter 9, right? The lab course also requires the lab manual, which is listed here, and this is what it looks like. This is also available on the Moodle site and for download. And there's also a free supplemental text, which looks like this here, that accompanies the lab manual. It's called a photographic atlas of microbiology. It's very useful if you're, like me, a really visual learner, because it's got lots of wonderful images and illustrations in it. And it is also available for a free download. All three of these texts are going to be posted in PDF format in the course materials module on the Moodle site, so you don't have to spend any money for these books. Please consult the syllabus and course Moodle site for course information, and do this regularly because things are always being added and updated. You're going to have very important things are going to be your news forum, which is on the first page, the course materials page. Your syllabus, course calendar, all of those are on the course material page. Guidelines and grading rubrics for the lab reports and your semester project reports. There's going to be some other writing resources in there having to do with formatting in APA format, how to cite references, things like that. Those are going to be very important for this class. Also on the Moodle site are all of the PowerPoint slides that accompany these videos, and your links for all of your quizzes, assignments, exams. All of your exam and project stuff is going to be posted on Moodle. They have their own modules. So it's not just the assignment link, but also other information to go with those. The videos are going to be posted on my YouTube channel, and they're at this link here. It's just www.youtube.com/c/Professor Sims. Please remember that these videos are designed to supplement and not substitute for your reading assignments. So that means you need to do the reading. And all of your reading is going to be at the end of these videos. It's also on the course calendar and the syllabus. The Moodle module for each lab includes a quiz. See the quiz links for due dates. Moodle is going to have, again, all of the information that you need regarding the quizzes, assignments, exams, everything. And all of the policies for grading, late assignments, missed exams, etc. can be found in the course syllabus, so please be sure to read through the syllabus. I have listed here office hours these are my traditional office hours. As of right now, I'm only doing office hours online, which may or may not change later in the semester. I'll be sure to let y'all know. But as of right now, we're doing office hours via Zoom conferencing. Also, you can ask questions by email or in the news forum on Moodle. Be sure to read and study all materials provided, like your textbook, procedures, your videos. Go through all of that stuff and take the quiz prior to performing lab procedures for any of your lab sessions. The quizzes are designed to make sure that you're ready to do the procedures. That is, do you understand why the experiments are being done? Do you understand what info they are meant to convey, how the procedures work, all of that kinds of stuff? It makes sure that you're actually getting the enhanced learning from doing the procedures rather than kind of learning as you go, because you're just not going to get as much from that experience as if you were prepared beforehand. Okay. If you can, you're going to want to print your procedures and the report templates beforehand so that you can keep them handy when you're performing your lab procedures. If you can't print them, that's okay. I recommend making detailed notes beforehand, though. You can have a laptop in your home kitchen lab, but I don't recommend it because sometimes things can get kind of messy. You don't want to mess up your laptop or possibly contaminate it. Having said that, not all of your lab procedures are going to be performed in a wet lab, environment. Some of them, in fact, fully half of the procedures are actually going to be simulations that you do virtually online. But you should take note of all of the procedural steps for those simulations as well. Record all of your results, all of your experimental data, especially results that are needed to complete the lab report templates. You do have templates for the lab reports, so if you look ahead to the results sections on those templates, you'll see exactly what you will need to report on ahead of time, which is helpful. And you want to utilize the expertise of your instructor and even your peers. You can ask questions on the discussion forum. You can use email to contact your instructors, attend Zoom office hours. You can even just post questions on the YouTube video, like the comment sections there. So, maybe there will be other people that will see your questions before I get to them or before Dr. Bhattacharya gets to them. Utilize all of those sources to ask questions. You don't have to be doing this all by yourself. Okay, so your lab kits, you're going to be coming to the university during the first week of classes. What we'll do is we'll have a pickup situation where we'll have your lab kits at the Biological Learning and Research building. You will come during a specified day. We'll set up some kind of an appointment system. You'll come to the parking lot and call in, and we'll bring out your lab kit for you. The lab kits you're going to be using at home in order to complete some of the lab procedures. The lab kits are going to include a fold scope, which is a neat little, very inexpensive handheld microscope. It's made out of like cardboard and paper, and it comes with a magnifying lens, and it couples to your cell phone camera. Very practical, very inexpensive, and very useful. You're also going to have a kit from Amscope that has materials in it for growing bacteria, for pouring petri dishes, nutrient agar plates. It has dyes in it for staining bacteria. There's some sterile cotton swabs and dropper bottles and things like that. You'll also have a slide box that has some glass slides that have been pre-made, and also some glass slides that are clean so that you can make your own slides. There's going to be other stuff in there like little UV flashlights, glow germ powder, lyophilized pond water specimen, things like that, transfer pipettes. So you'll get your kit, and with your kit you'll have a detailed list of everything that's in there. And for the lab procedures that require supplies from the kit, it will say specifically on there exactly what you will need from the kit. So let's talk about safety. Safety can be kind of challenging, especially from an online lab where you're doing everything at home and are not supervised. So what has happened is that the on-site micro lab is a BSL-2 lab where we work with pathogens. But for the online lab, it's been retooled such that it's a BSL-1 lab. So BSL means biosafety level. Biosafety level 1 is the lowest level of biosafety, level that applies to agents that do not ordinarily cause human disease. However, please keep in mind that all specimen that you will handle, culture, or work with should always be considered a potential pathogen. So you want to be careful. Even though it's a BSL-1 lab, you still want to treat it as if you're working with pathogens. Also, some of the reagents are harmful, such as the staining dyes that you're going to be using. These are classified as potential carcinogens. Carcinogen, of course, being a cancer-causing agent. So you're going to want to be very careful with those. When performing your lab procedures at home, be sure to follow all safety rules. The safety rules are going to be posted in the course materials module on Moodle and also included within your lab procedures. Students must complete a mandatory safety quiz before executing any of the lab procedures. This is going to be in the Introduction to Micro Lab module on Moodle, and it is due during the first week of class. Although you will be performing your lab procedures at home, most likely in your kitchen, it's important to keep all lab specimen and reagents away from food. In fact, if it's available, a bathroom sink with an adjacent counter that is physically separated by a door or a wall from your toilet would be an ideal location for your at-home lab. You don't want the lab stuff too close to a toilet though, because toilets release aerosols containing E. coli and other microbes when they're flushed. And that's true even if you flush with the toilet lid closed, by the way. When you're incubating specimen, you want to store those plates in a warm, dark place, again, away from food. You want to tape the plates closed to secure the lids. Inoculated plates, and sterile plates too, can be stored in the fridge, but you want them to be in an airtight container. So something like a Ziploc bag or a Gladware, like one of those plastic containers with the lids, those would be fine. You want to disinfect your counters or any surface before and after performing any lab procedures. So suitable disinfectants include Lysol, Formula 409 is really good, and obviously bleach, diluted bleach. You want to wait at least 1 hour after disinfecting those counters before you prepare any food on or even near those counters. Personal protective equipment, PPE, is recommended. I am aware that it may not be available at this time for all students. So at the very least, you want some form of protection for your eyes, hands, and your torso. Eye protection is going to be in the form of some kind of safety glasses or goggles. Hand protection includes gloves. If you don't have access to nitrile or latex gloves, you can use those rubber dishwashing gloves. Those will work as well. Torso protection can be in the form of a lab coat, ideally. But you can also use a long-sleeved scrub shirt or really any long-sleeve shirt. The main thing is that you have to have your entire torso and your arms covered. And whatever you use to do that, you only want to use that when performing your lab procedures. So don't wear the same top that you were wearing to do slide staining or something like that, and then turn around and make lunch or something, okay? You want to change out of that top after you're done doing your lab procedures. Keep in mind that even though we are working with BSL-1 microbes, some experiments require you to culture species that are unknown. So you should treat every specimen as a potential pathogen. So we're going to be doing some hand washing experiments. We're going to be looking at ubiquity of microbes. And for those, you're going to be culturing things that I don't know what it is. I don't know what's on your hands. I don't know what's on the fomite surfaces that you swab. So they could be pathogenic, and therefore you want to treat everything. Just treat everything as if it is pathogenic. So you'll just be extra careful with them, okay? And finally, we'll talk about disposing of things. So after you use your plates and your slides, you'll have to dispose of them properly so you're not endangering yourself or the environment or anything like that. The key here is to decontaminate items before you dispose of them. As long as you're not collecting samples from the bathroom, okay, where there's definitely pathogenic bacteria, it should be safe to decontaminate and dispose of the specimen plates and the slides at home in your regular garbage. Before you attempt to dispose of your petri dishes, you first have to kill the bacteria that's on them. And basically the only way to do this is with bleach. So you're going to protect your hands and your eyes, and you pour just a small amount of bleach onto the agar, onto the plate. You let that soak for 30 minutes. Then you're going to seal the plate either with plastic wrap or you're going to put it in a Ziploc bag, but it has to be totally sealed up before putting that in the trash. For the slides, you want to soak your slides in water that contains either bleach or you can use an antimicrobial dish soap. and let that soak for at least 1 hour. Also, it's best to put the glass slides after they've been contaminated, put them in some kind of hard plastic container, or you can wrap it in cardboard or bubble tape, something like that, before disposing it in the trash. And you want to do that because obviously these are sharps and we're not putting them in sharps containers. So you can make your own sharps container just by putting it in some kind of plastic jug or something like that. Or you can wrap the slides in something that will keep them from breaking when they're in the trash, like cardboard or something like that. Finally, don't forget. Okay, so thank you guys for watching. As always, don't forget to do the reading. Check the description below this video for more videos related to these topics. And of course, you can leave questions for me here in the comment section. And I'll see you for the next one.